Okay, next up is apoptosis. So we're going to talk about the causes of apoptosis, some of the morphologic and biochemical changes that you see in apoptosis, and then finally the mechanisms. So we've already touched on apoptosis a little bit. So just a quick overview. So again, this is a pathway of cell death that is induced by a tightly regulated suicide program. So this is sometimes called programmed cell death. Now, cells that are destined to die are going to activate intrinsic enzymes that are going to cause degradation of the cell's own parts. So the nuclear DNA, the nuclear proteins, and all the cytoplasmic proteins. And then as this happens, the cell is going to start to break up. And so it's going to essentially like fall apart, but in a very controlled manner. So these are apoptotic bodies. So these are little fragments of the cell that are going to come off. These are called apoptotic bodies, and these are going to be cleaned up very rapidly. So whatever's left of the cell and all the apoptotic bodies are cleaned up rapidly, and that is why in apoptosis you're not really going to see inflammation, whereas in necrosis you do. So that's one of the main differences between the two that's very high yield. So in this picture, you can see this is what an apoptotic cell looks like. So this is a part of the liver, and so this is a small focus of inflammation in the liver with an apoptotic cell. Now, importantly, I'm pointing this out because this is something called the bystander effect. So I just said that you shouldn't see inflammation with apoptosis, and now I'm showing you a picture where I'm saying there's a lot of there's a lot of inflammation and apoptosis. So this is just because this cell didn't become apoptotic and rupture and cause the inflammation, but instead the inflammation was already there. This was causing an adverse environment for this hepatocyte, and so because the hepatocyte was agitated and irritated, it initiated apoptosis. So the inflammation was there first. So if you see something like this where you're seeing a lot of inflammatory cells, but you also see apoptotic cells, don't automatically assume, oh, that must be necrotic instead, just because you see the inflammation. So again, this is just something called the bystander effect, and I just wanted to point that out in case you're looking at things that have some inflammation. So this here where the arrow is pointing to, oh, red is not going to work. Let's do pen maybe? Laser color. Ooh, fancy. Okay, here we go. So this that the arrow is pointing to, this is an apoptotic cell. So you can see it's very eosinophilic. We've talked about that. It's swollen. It's much bigger than the surrounding cells. And you can see that the nucleus is fragmented here. So this is an apoptotic cell surrounded by inflammatory cells. So bystander effect, just wanted to mention that. Now, the causes of apoptosis, so we said that this is programmed cell death. So this is usually like a normal physiologic thing, but like we just saw with that bystander effect, it can be pathologic too. So there's some physiologic causes and there's some pathologic causes. So some examples of normal physiologic causes of apoptosis are embryogenesis. So during embryonic development, some cells are going to have to undergo apoptosis. That's just a normal process. Another example is involution of hormone-dependent tissues when you remove that hormone. So an example of this is endometrial cells during menopause. So, or also ovarian follicles during menopause. So those are going to they're going to get atrophied, and so they become atretic follicles. And so during menopause, you're going to have decreased levels of hormones that normally stimulate these tissues, the endometrium and the ovaries. And so if you decrease the amount of hormone, those cells are naturally going to undergo apoptosis. That's a normal physiologic response. Another example from female would be a breast that is no longer lactating. So during pregnancy, the breasts are going to get bigger. So we talked about that in the cell adaptation lecture. And then after the woman is no longer breastfeeding, those are going to then, a lot of the cells are going to undergo apoptosis so that the breast can go back to quote unquote normal. And then another example from males, after castration, if you have to have a testicle or both testicles removed for whatever reason, that you'll have a decreased level of testosterone, and so that can cause atrophy of the prostatic tissue. So those are just some examples of how this would be just a normal physiologic response. having to do with hormones. And then another example is if you're losing cells in a cell population that's normally proliferating a lot to maintain homeostasis, because if these cells are constantly proliferating but none of them are being killed off, then you're going to have too many cells. So in order to maintain that normal homeostasis, you have to have some that are reproducing and some that are, degenerating. So an example of this would be like the lymphocytes in bone marrow. So these immature lymphocytes in bone marrow are constantly, the number is constantly increasing. So some are going to have to undergo apoptosis to maintain that homeostasis. Another example would be your intestinal cells. So epithelial cells in the intestinal crypts. These are going to have to undergo apoptosis, some of them, in order to maintain that proper amount. Another example of a physiologic cause would be death of host cells that have done their job. So an example for this would be neutrophils. So neutrophils, what are those normally used for, right? That's going to be your acute inflammatory cell. So after acute inflammation, they've already done their job. And so those can undergo apoptosis because they've already done what they needed to do, and now they need to just go away, essentially. And then the last example of physiologic atrophy or apoptosis would be elimination of cells that are potentially self-reactive. So in the thymus, you have these T cells that are maturing, and as this is happening, you have selection. So you have positive and negative selection of T cells. And so the body's essentially going to kill off any of these T cells that could be self-reactive. So here's an example of that. So this is in the thymus, and you can see the thymocytes in there. So like I said, normally these are going to undergo the positive and negative selection to produce T cells that definitely will not have any auto-reactive parts. So if there's any T cells that might recognize the self MHC, those would be negatively selected and essentially undergo apoptosis. So this is shown in this picture. You can see that, this is actually really pretty. So you can see, let's change color. Now red will work. So this right here, this is something that we call a tingible body macrophage. And so this is a macrophage that has eaten up a couple of these dead and dying lymphocytes. So this would be an apoptotic lymphocyte, and this whole thing is a macrophage that has eaten it up. And so this is actually from a mouse. This isn't a human tissue, but I thought this was a really good example of this normal physiologic cause of apoptosis. So this is in the thymus, and again, these cells are being killed off, in a controlled manner because they might be self-reactive. So those are the major physiologic causes of apoptosis. Now, you can also get a pathologic cause of apoptosis, right? So one example of this would be DNA damage. So DNA damage is obviously going to be damaging to the cell. And like we mentioned in a previous lecture, we do have some mechanisms to fix DNA damage, but only to a certain point. So if it gets so bad that the cell has to be killed, it's going to undergo apoptosis. And another example would be accumulation of misfolded proteins. That's kind of similar to the DNA damage, and then cell death in certain infections. So this is particularly viral infections like HIV or something like viral hepatitis that can cause cell death. And lastly, pathologic atrophy after duct obstruction. So if you're blocking a duct, that can affect the downstream or the upstream cells of that duct. And so this can happen in glandular tissues like pancreas and parotid. It can also happen in the kidney. So if those cells are being damaged because they either have an increased amount of enzymes in them, so say you block the pancreatic duct, those enzymes in the pancreas are going to be damaging to the pancreas, and so those cells can undergo apoptosis. Now, there probably would also be some necrosis, but you can have some just normal regulated apoptosis as well. So those are just the general causes of apoptosis, and as we get into the rest of general and then systemic pathology, we'll talk about this with each organ system. So this is just kind of an overview. Keep that in mind. Now, what are the different morphologic and biochemical changes that you're going to see during apoptosis? And a lot of these we've already talked about. So the cells are going to shrink, the chromatin inside the cell is going to condense and stain darker. You're going to have some cytoplasmic blebbing as the cell undergoes apoptosis. Part of it is going to come off as these little apoptotic bodies, and then those apoptotic bodies are going to be eaten up by macrophages that come in and clean it up really quickly, like within a few minutes. And that's why you don't necessarily have inflammation with apoptosis. So here's a couple examples of what these look like. So this is another section here. This looks like pancreas, and so this is super zoomed in, but these would be normal cells, and you can see that you have these arrows are pointing to these weird looking cells. So these do not look like the surrounding cells, so that's because these are undergoing apoptosis. So you can see that the nuclei have condensed. They're a little bit darker staining, and you can also see them fragmenting. So this one here, you can see it's super dark. You can kind of see it's beginning to fragment. Same thing with this one over here, whereas this one, it has almost completely fragmented the nucleus in there. You can also see the cytoplasm is also sort of condensing. It's also becoming a lot more eosinophilic. So overall, just the cell in general has shrunk compared to the surrounding cells. It is more eosinophilic, and that nucleus is more basophilic and starts to fragment. Here's another example. So this one looks like it's from liver, and so again, here these would be your normal hepatocytes. So like this is a nice normal hepatocyte. You can see that there is some fatty change in here, but that is not normal, but fairly common in the liver. And then you can see this would be an apoptotic cell here. So again, the nucleus, the chromatin has condensed, the nucleus starts to fragment, and then overall that cytoplasm is more eosinophilic, and in general, the whole cell has kind of shrunk. Same thing here. So this, you can see that these cells that all of these arrows are pointing to, they're all kind of in different phases of apoptosis. So this one, there's no nucleus anymore. Same thing with this one. Those are already pretty much entirely fragmented. But then here you can see the nucleus is fragmented, and then this one here, you can see it's starting to condense and stain darker, and the cytoplasm is starting to become more eosinophilic. So this would be like the end of apoptosis. It's about to be eaten up by a macrophage probably, and then this one is early stage of apoptosis. So those are some of the morphologic changes. Now, what are the mechanisms? How does this actually happen? So the main underlying thing in apoptosis is that you are activating those caspases, which we talked about in a previous lecture. So a marker for cells undergoing apoptosis would be presence of cleaved and activated caspases. And so it happens through this process. It has two phases. So the first phase is going to be initiation phase. And this, within the initiation phase, there's two main pathways. So you can either go through the mitochondrial or the intrinsic pathway, or you can go through the death receptor or the extrinsic pathway. So that's the initiation phase. And then after the initiation phase, both of these are then going to move on to the execution phase. So here in the initiation phase, this is how apoptosis begins, and that's going to involve the activation of these caspases, okay? And then when this initiation part finishes, you're going to move on to the execution phase, which is also going to activate some more caspases. And so each phase is going to have specific caspases that are activated and cleaved. So we're going to go through this. If you're reading about this in the book, it can get a little bit wordy and confusing, and so I tried to split it up in a manageable way. So I made some doodles. Hopefully this helps. If you find that it's more confusing and you'd rather read about it, that's also fine. I just thought that this might make it a little bit more simple for you. So we're talking about the initiation phase, and we're going to talk about the two pathways separately. So the first pathway is that intrinsic pathway. So this is happening in the mitochondria. That's why it's also known as the intrinsic pathway, because it's inside the cell. Now, this is the major mechanism in pretty much all mammalian cells. So basically what happens is you have increased permeability of the outer membrane of the mitochondria. And that is going to cause release of those pro-apoptotic molecules in the cytoplasm. So do you remember what the main one that I said in a previous lecture was? It's cytochrome C, right? And so the cytochrome C is going to be released, and that is going to cause basically a downstream pathway. So this whole mechanism, this mitochondrial or intrinsic pathway, is controlled by these proteins that are in the BCL2 family. So there's three main types of these cells that are, proteins that are in this BCL2 family. They can be split into three groups. And so the first group, these are anti-apoptotic. So these are essentially protective against apoptosis. And two of the main ones that I've listed here are BCL2 and BCL-XL. So both of these, they're in the outer membrane of the mitochondria, and their job is to keep it impermeable. So these, again, if they're keeping that membrane impermeable, they're keeping it controlled and normal, so anti-apoptotic. So these are essentially preventing leakage of cytochrome C. The second group of these proteins are pro-apoptotic. So the two main ones are BAX and BAC, and I definitely want you to know these. But BAX and BAC, these are also located on the outer membrane, and when these are activated, they do the opposite. So they're going to increase permeability of that outer membrane of the mitochondria. And so if you're increasing the permeability, that's going to cause that release of those pro-apoptotic molecules, which are your cytochrome C. And then you have this third, I forgot I put this drawing in. So here you can see where they're actually located. So BAX and BAC are those pro-apoptotic molecules, and they're on the outer membrane of the mitochondria. So this is my version of a mitochondria. Now the third group are the sensors. So the sensors are essentially sensing cell stress and damage, and they help to regulate the balance between those two other groups, so the anti and the pro-apoptotic groups. And so if these sensors sense that there's some sort of cell damage, they can decrease the stimulus to the anti-apoptotic proteins and increase the activity or stimulate the pro-apoptotic proteins. So the main one that I want you to know is BAD. So go ahead and circle that one. You should know BAD. Okay, so those are the three main groups of the proteins that control this whole pathway. But you also need to understand the role of each. So we're still talking about this intrinsic pathway. So here's where the doodling comes into play. So this is that same picture we just saw. So we have the BAX and the BAC. Remember, these are on the outer membrane of the mitochondria, and these are pro-apoptotic. So if the cell is damaged, those sensor proteins, so like your BAD, those are going to activate BAX and BAC. And then once these are activated, that would cause this to become permeable, right? But remember, you still have those anti-apoptotic proteins as well. And so you're going to have a decrease in survival signals that are normally stimulating these anti-apoptotic proteins. And so if you have a decrease in these survival signals, that is going to cause a decrease in BCL2 and BCL-XL activity and stimulation. So if you're decreasing the anti-apoptotic proteins and you're increasing the activation of the pro-apoptotic, those are both going to have a net effect of causing the release of these proteins, so cytochrome C, into that cytoplasm. And so that is going to initiate a caspase cascade, okay? So when the cytochrome C is released into the cell cytoplasm, it can then come out and it's going to be released through the BAX and the BAC because these have now made that outer membrane permeable, right? So the cytochrome C is released into the cytoplasm, and then it's going to leave through the membrane because of the activity of these pro-apoptotic proteins, BAX and BAC. Now, once it gets out, into the cytoplasm, so it's leaving the mitochondria, it's going into the cytoplasm here. Once it gets out, then it's going to bind to something called APAF1 or APAF1. Now, what this stands for is apoptosis activating factor 1. I know that's probably hard to read, so that's APAF-1, and that is your apoptosis activating factor 1. And once cytochrome C binds to the APAF1, that forms something called the apoptosome. Now, the apoptosome is going to bind caspase 9, and this caspase 9, this is the critical initiator of the caspase cascade within this mitochondrial pathway. So that is definitely something that you should know. So the apoptosome binds the caspase 9. That is going to cause cleavage of additional caspase 9s. And so it's essentially this auto-amplification of all of these caspase 9s. As all of these are becoming cleaved, that's what activates them. And so this is a cascade of caspase activation through cleaving of all of these caspases. And then you're going to go to the execution phase, which we'll talk about in a couple of slides. So all of this was the intrinsic or the mitochondrial pathway, which is in the initiation phase, okay? But then the second pathway that you can initiate apoptosis is through this extrinsic pathway, which is your death receptor pathway. So this is going to be regulated by these death receptors in the plasma membrane, and these are all in the TNF alpha family, all of these death receptors. So this is going to be initiated by the engagement of these plasma membrane death receptors, and the example we're going to use is FAS. So you should probably know FAS. So FAS is expressed, or FAS ligand, so what FAS is binding with, that's going to be expressed on T cells that recognize self-antigen. So think back to when we looked at that picture of the thymus and how some of those cells were undergoing apoptosis. That's what we're talking about. So the FAS ligand is expressed on these T cells that recognize self-antigens. Then the FAS ligand is going to bind to FAS, and that is going to cause a cytoplasmic death domain to form a binding site for FADD, okay? So here we have the FAS death receptor binding with the FAS ligand, and that is then going to initiate activation of procaspases 8 and 10. Those are the ones that are common in humans. And as you bind to this, it's going to cleave that and form the procaspase, then becomes an activated cleaved caspase, so caspase 8 and 10. Those are then going to also cause activation of Baxan back. Then the cytochrome C is going to be released into the cell cytoplasm, right? So if this is the plasma membrane, this is the mitochondria, all of this cytochrome C is now inside that cytoplasm. And then it's essentially the same as the other pathway, right? So we're releasing cytochrome C. Cytochrome C is going to bind to APF1 to form that apoptosome. And then the apoptosome is going to bind to caspase 9. And so now we're at the same part that we were at before. So these are just the two different pathways that we can initiate this apoptosis. So the main takeaways is that the intrinsic pathway is going to be through caspase 9, and the death receptor or the extrinsic pathway is going to be through the use of caspase 8 and 10. And think of why this is intrinsic versus extrinsic. So the intrinsic pathway is because you're having some sort of damage to that mitochondria, and that's going to then cause permeability of that membrane to then release the cytochrome C into the cytoplasm. Versus the extrinsic pathway, this is something coming from the outside of the cell. So because you have that plasma membrane receptor, it's coming and it's interacting with the outside of the cell. That is then going to initiate activation of caspases 8 and 10, which are then going to cause release of cytochrome C from the mitochondria into the cytoplasm. But the net result is the same. The net result is that you're releasing that cytochrome C. So after that initiation phase, we're then going to move on to the execution phase. So this is mediated by the executioner caspases, which are 3 and 6. And so these caspases are going to degrade the structural components of the cell and promote that cell fragmentation. So basically what happens, no matter which pathway you're doing, whether you're coming from the extrinsic initiation and coming through here to activate caspase 8 and 10, the ultimate result of that is to release cytochrome C. If it's the intrinsic pathway where you have damage here, that's going to cause release of cytochrome C. So both of those are going to cause activation of caspase 9. Caspase 9 can then initiate activation of caspases 3 and 6, which are your executioner caspases. But also, something that you should know is that the extrinsic pathway over here, caspase 8 and 10, also stimulate the the activation of caspases 3 and 6. So both of these are ending in the same way. So the extrinsic pathway is going to cause activation of caspase 8 and 9, and then indirectly also cause initiation of caspase 9. All of that is going to eventually cause activation of the 3 and 6, which are your executioners. But the extrinsic pathway also directly stimulates activation of caspase 3 and 6 as well. And basically, when you initiate the activation of these caspases 3 and 6, because these are your executioner caspases, that is going to lead to cell death. So here's kind of the combination, both on one screen. Here's the difference between the two pathways. So ultimately, they have this common effect of activating these caspase 3 and 6, which are your executioners. It's just that the mechanism of getting there, the initiation, is a little bit different. So you should be able to explain this either in words, or you can like draw it out, however you prefer to do it. Definitely go through this and make sure you know the difference between these two pathways and the similarities between these two pathways. But ultimately, the whole point is to start to degrade this cell. And once this cell is degraded, you're going to want to remove it very quickly in order to prevent that inflammation, right? So the final part of the mechanism of apoptosis is removal of those dead cells. So as the cells are dying, they're going to secrete these soluble factors that recruit phagocytes. And so these are essentially like eat me signals. So they're sending out these signals that are saying, hey, come eat me, clean me up. And so that is going to cause these phagocytes to come in. So mainly like macrophages, those will come in and they'll clean these up within minutes. And that's why you don't have inflammation. And another thing is that these dying cells can actually be covered in proteins of the complement system, and that can help them get cleaned up as well. So here you can see that these are so beautiful. So these are more tingible body macrophages. So here, all of these, so where's my arrow? There we go. So like this here, that all the arrows are pointing to some of them, but these are tingible body macrophages. So here's your macrophage. This is the macrophage cytoplasm, and all of this is dead or dying apoptotic cells within that macrophage cytoplasm. So these macrophages have come in. Here's another one. All of these are dead and dying cells within the macrophage. How cool is that? So the macrophage will come in and it will eat up all of these little cell fragments. And then over here, it looks like this is an apoptotic cell, and you can see that it has a little, what is it called? An apoptotic body coming off of it. So that's pretty cool too. Here's another example of another tingible body macrophage. So here, these are fragments of dead and dying cells, and they're all within the cytoplasm of this one macrophage. So tingible body macrophage, you should know the name of that. And then lastly, can you tell I like these? Here you have some dying cells. So this would be an apoptotic cell. And then you can even see some of them, like this one, you can see it's all fragmenting. This is also all fragmenting. These are beautiful examples of apoptotic cells. And these honestly can be kind of hard to identify histologically, because if it's a necrotic area, it's very obvious because there's tons of inflammation, and inflammation is very obvious under the microscope. But if you're looking for apoptotic cells, it's usually a little bit less obvious because they aren't surrounded by tons of inflammation. So this is a really good example of a physiologic apoptosis, because this is a section from a mammary gland from, actually I think this is from an animal, but this is basically the lactation was stopped for 5 days, and so now you can see all of these different cells, the epithelial cells within that mammary gland are going to start to undergo apoptosis. So that's a beautiful example of apoptotic cells. But it's hard to find good pictures of these because, again, they're harder to identify under the microscope. You have to really look for them. But there are certain areas, like in a mammary gland that was lactating and is undergoing apoptosis, or in the thymus. Like, you can find good examples of it. So that is apoptosis. So I want you to know the mechanisms for sure, and then definitely know some of the morphologic changes that you're going to see in apoptosis.